So I want you to imagine, just for a second, trying to swim through uh, a massive pool that has been entirely filled with thick, dense gelatin.
Oh, wow. Okay.
Right. I mean, most things would hit the surface and immediately get bogged down.
Yeah, absolutely. If you are a standard microscopic bacterium, trying to paddle your way through that kind of viscous environment using a little external tail, you're going nowhere. The fluid dynamics at that microscopic scale mean the gelatin basically acts like a solid wall.
Your tail just gets totally tangled.
Exactly. You're stuck.
But what if you had a drill built directly into your body? You wouldn't need to paddle at all. You could just bore straight through.
Yeah.
And looking at the stack of sources for this deep dive today, it becomes incredibly clear that this bizarre corkscrew body plan is the key to unlocking centuries of medical mysteries.
It really is.
Today, we are exploring a truly fascinating bacterial phylum known as the Spirochetes. We're going to unpack the biology, the complex history, and, the very real modern-day consequences of this unique microscopic family. And we'll be specifically focusing on its two most infamous members, the bacteria behind syphilis and the bacteria behind Lyme disease.
And I think it's important to note right up front, the documents we are analyzing highlight that this isn't just a standard biology lesson.
No, not at all.
It's a story of medical rivalries, intense historical debates, and fundamentally how a very specific microscopic shape dictates exactly how these diseases infiltrate and attack the human body.
Yeah, the shape is everything. To get a clear picture of what we're talking about, I want you to keep a core visual in your mind as we go through this. If most swimming bacteria in the world are like speedboats, using outboard motors, which are these little external tails called flagella to paddle through the water, Spirochetes are entirely different.
They're a completely different paradigm.
Right. They are built like submarines with an internal corkscrew designed to literally bore through dense, impenetrable materials. The mechanics of how a single-celled organism builds a corkscrew into its own body are just astonishing to me.
It is a profound piece of biological engineering. I mean, most swimming bacteria have those whip-like tails on the outside of their bodies freely spinning in the surrounding fluid. But the Spirochete took a totally divergent evolutionary path. Its flagella are tucked entirely inside its own structure.
Wait, really? Like completely inside the cell?
Yes, exactly. They run the entire length of the cell, sandwiched securely between an inner cytoplasmic membrane and an outer membrane sheath. In the scientific literature we looked at, you'll see these structures referred to as periplasmic flagella, endoflagella, or axial filaments.
Okay, so because they are trapped inside that outer layer, they must change the entire movement dynamic, right?
They do. When those internal flagella rotate, they don't just spin fluid out of the way like a normal tail would. The torque they generate actually twists the entire outer cell body into a tight helical coil.
So the whole bacterium becomes the corkscrew.
Exactly. It converts rotational force directly into forward thrust. It's a highly sophisticated mechanical solution to a mechanical problem. Spirochetes did not evolve to live in open, watery environments where an external tail works perfectly fine.
Right, they specialize in thick environments.
Yes, they colonize thick, viscous landscapes. We are talking about human connective tissue, dense mucosal surfaces, and cerebrospinal fluid. An external tail would just get caught in that biological mesh. A corkscrew, however, can penetrate.
Which perfectly explains the gelatin pool analogy. Now, before we get into the specific diseases, the sources highlight a major taxonomy trap that really trips people up. Spirochete is not a single disease.
No, it's not.
It is an entire bacterial phylum. People constantly use the word incorrectly in casual conversation, saying things like, oh, they found the Spirochete in the patient, using it as a synonym for just the syphilis bacterium or just the Lyme bacterium, but it encompasses a massive grouping.
It's a huge family tree.
Yeah, it includes treponema, which causes syphilis, borrelia, which causes Lyme disease, and another genus entirely called Leptospira, which causes leptospirosis.
Right. The term describes the shape and the locomotion strategy shared by that entire branch of the bacterial family tree.
But looking at the history of taxonomy, classifying microorganisms purely by their shape seems incredibly risky to me. I mean, if we look at the animal kingdom, that is equivalent to calling a dolphin a fish just because it has fins and swims in the ocean. Shape does not always equal shared ancestry.
That's a great point.
I would think early microbiologists would have worried that a bunch of unrelated bacteria just coincidentally arrives at the same corkscrew shape because it was, mechanically useful.
And you'd be right to think that. For most of the 20th century, that was actually a massive concern in microbiology. Scientists wondered if this was just a case of convergent evolution. Spirochetes were grouped together almost entirely based on what scientists could see under the microscope, that distinct coil shape and that writhing, twisting movement.
So they didn't really know for sure if they were related.
Not definitively. The rigorous genetic proof didn't arrive until the 1980s and 90s, when genetic technology finally caught up. Researchers began using 16S rRNA gene sequencing.
Which is essentially a way of reading the molecular clock of a bacterium to trace its lineage, right?
Exactly. The 16S RNA gene mutates at a very slow, predictable rate, making it perfect for tracing ancient evolutionary relationships. When they sequenced the Spirochetes, the genetics confirmed that this was not a dolphin and fish situation.
Oh, wow. So they actually were related.
Yes, the sequencing proved that Spirochetes genuinely are a distinct ancient evolutionary lineage. They are a monophyletic group, meaning they all share a single common ancestor.
That's incredible.
Yeah, so the classification shifted definitively from things that happen to look like corkscrews to a deeply related family that looks like corkscrews because of a shared genetic heritage.
Okay, so knowing what these bacteria actually are mechanically, the journey to finding them is a wild ride through medical history. The sources detail a timeline spanning over a century, highlighting two wildly different methods of scientific discovery. The initial identification didn't even happen in a medical lab.
Right, it was much more humble than that.
Yeah, in 1835, a German naturalist named Christian Gottfried Ehrenberg was looking at pond water and sewage under early microscopes, just cataloging random microorganisms.
He observed these thread-like twisting organisms and, well, he needed a descriptive name. Combining the Greek words spera, meaning coil, and chate, meaning hair, he coined the term spirocheta, literally, coiled hair.
Coiled hair in pond scum is a very humble beginning for a biological machine that causes so much medical chaos. But the narrative shifts dramatically when we look at the first big medical discovery, syphilis.
It's a completely different vibe.
Completely. This happened on March 3rd, 1905, at the Charité Hospital in Berlin. The zoologist Fritz Schaudin and dermatologist Erich Hoffmann were examining material from a syphilitic lesion known as a chancre, and this was an intense, high-pressure race. The scientific community was desperately hunting for the cause of syphilis, and the prestige of finding it was immense.
The challenge they faced was optical, though. These bacteria are incredibly thin and practically transparent. A standard light microscope looks right through them, which is why so many other researchers had failed to spot them.
So how did they finally see them?
Schaudin and Hoffmann succeeded by using dark field microscopy. This is a technique where you illuminate the sample from the side against a pitch-black background. Instead of shining light directly through the bacteria, you look at the light scattering off their edges.
Oh, that makes sense. It is exactly like seeing dust motes floating in a single shaft of sunlight in an otherwise dark room.
That's a perfect analogy, yeah. In that scattered light, they finally spotted thin, tightly coiled organisms writhing through the sample. They named it Spirocheta pallida because of its pale, faintly staining appearance.
Though today it is known as treponema pallidum.
Right. And Schaudin made this monumental identification, but he tragically died just 3 years later at the age of 34 from an amoebic infection. He completely missed the medical revolution his discovery sparked.
Wow, that is incredibly tragic. It was a lightning-fast, high-stakes discovery, and then he's just gone.
Yeah, and the identification of the Lyme disease spirochete, which happened decades later between 1975 and 1982, offers a complete contrast to that kind of frantic pace.
Definitely. The Lyme discovery didn't start with a scientist in a lab at all. It started with an observant mother in Connecticut named Polly Murray. She noticed an unusual, really alarming cluster of what looked like juvenile rheumatoid arthritis in her town of Lyme.
Kids and adults were suffering from severe joint pain, fatigue, and these strange rashes.
Yeah, and she meticulously documented the cases and sounded the alarm, eventually bringing rheumatologist Alan Steer from Yale in to investigate this mysterious Lyme arthritis.
But for years, the actual cause remained a total mystery. The breakthrough only happened when an entomologist named Willy Burgdorfer got involved. Burgdorfer was meticulously dissecting Ixodes ticks from Long Island and Connecticut, but, and this is the crazy part, he was actually looking for the cause of an entirely different illness. Rocky Mountain spotted fever.
Right, he wasn't even looking for Lyme disease. He was doing completely unrelated tick surveillance.
Exactly. While examining the tick's midguts under the microscope, he spotted the familiar writhing shape of Spirochetes. Recognizing the potential connection, He took blood serum containing antibodies from the Lyme patients and tested them against these newly found tick spirochetes.
And it was a match.
A perfect match. The antibodies reacted strongly. The mystery was solved, and the bacterium was named Borrelia burgdorferi in his honor.
The contrast there is incredible. You have an action movie race against the clock in a Berlin hospital for syphilis, and then a methodical detective novel for Lyme, where the investigator actually trips over the crucial clue while looking for something else entirely.
It really highlights how unpredictable science can be.
For sure. Now, those discoveries laid the groundwork, but looking at the clinical data in our sources, the real marvel is how these corkscrews operate once inside the human body. It all comes down to extreme biological specialization and timing.
Let's start with mechanical penetration. Because they are corkscrews, treponema pallidum can burrow directly through intact mucosal epithelium during sexual contact. It literally drills its way into the tissue.
Which is terrifying.
Yeah. And similarly, when an infected tick bites you, Borrelia burgdorferi uses that same corkscrew motion to migrate outward from the bite site, weaving through the dense layers of your skin. That physical outward migration of the bacteria is exactly what causes the classic bullseye rash, known clinically as erythema migrans. The bacteria are literally spiraling away from the center.
That makes the visual of the rash make so much more sense. Now, the biology of the syphilis bacterium, treponema pallidum, is particularly mind-boggling. The sources note it has a tiny genome, just 1.14 million base pairs. To put that in perspective, a standard E. coli bacterium has over 4 million base pairs.
Right, it's missing a lot of parts.
Treponema lacks the biological machinery to synthesize its own amino acids or its own lipids. It basically has to scavenge everything from the human host. It seems entirely counterintuitive that a bacterium missing major parts of its own internal factory is so devastatingly good at infecting us.
It feels like a contradiction, for sure, but its extreme parasitism is the foundation of its success. It has stripped away everything it does not absolutely need, dedicating all its available energy to one highly specialized task: exploiting human biology and evading the immune system.
It just relies completely on us.
Yeah. It doesn't need a massive genome to survive in the wild because it never lives in the wild. Its entire universe is the human body.
It is so dependent on us that for over a century, scientists could not grow it in a lab dish. They had to maintain the bacteria by constantly infecting live rabbit tissue just to study it. It wasn't until 2018 that researchers Edmondson, Norris, and their colleagues finally figured out how to culture it in vitro, using a highly specialized co-culture with rabbit epithelial cells.
And because treponema pallidum is so specialized for hiding and slowly scavenging within human tissues, the disease it causes unfolds in complex, time-separated stages. This elusive nature is exactly why syphilis earned the historical nickname, the Great Imitator.
Right. The primary stage usually appears about 3 weeks after exposure. It presents as a painless ulcer, or chancre, right where the bacteria drilled into the tissue. Because it is painless, it often goes completely unnoticed.
Weeks or sometimes months later, the secondary stage begins. By then, the bacteria have disseminated throughout the bloodstream. This typically causes a diffuse rash, notably appearing on the palms of the hands and soles of the feet, which, by the way, is unusual for most rashes, and it's often accompanied by fever and swollen lymph nodes.
But then it just goes away, right? After the secondary stage clears up, the infection enters the latent period. This can last for years or even decades with absolutely no outward symptoms. A major misconception here is that the absence of symptoms means the infection has run its course and disappeared.
That is a very dangerous misconception. The bacteria are still very much alive, they're just hiding deep in the tissues. In about 15 to 30% of untreated cases, the disease eventually roars back as tertiary syphilis.
And that's when things get really bad.
The damage at this stage is catastrophic. It causes massive cardiovascular destruction, such as aortic aneurysms or neurosyphilis, which leads to severe progressive dementia, blindness, and paralysis. Before the widespread use of antibiotics, tertiary syphilis was a leading cause of patients being permanently institutionalized. It mimics dermatologic, cardiac, and psychiatric disorders perfectly.
Well, syphilis is built for immediate invasion upon contact. The Lyme bacterium, Borrelia burgdorferi, operates on a completely different timetable. Its invasion is dictated entirely by a tick's biological clock.
Yes, Borrelia resides in the midgut of the ixode's tick. When that tick attaches to a human for a blood meal, the bacteria do not instantly flow into the bloodstream.
That's a huge relief, honestly.
Definitely. They must first detect the influx of warm blood, detach from the gut wall, migrate up into the tick's salivary glands, and then pass into the human host.
And that migration process takes roughly 36 to 48 hours of continuous tick attachment and feeding. This busts a huge misconception. You cannot get Lyme disease the second the tick touches you, or even if it is attached for just a few hours. Doing a prompt tick check after a hike genuinely works because you are physically beating that biological migration clock.
It's the best defense we have. There is another significant misconception regarding Lyme testing, too. If a patient is bitten by a tick and goes to the doctor the very next day, an early negative Lyme test does not rule out the infection. Standard Lyme serology tests do not look for the bacteria itself. They look for the antibodies the human immune system produces to fight the Spirochete.
And the body needs time to build that detectable antibody response. Testing too early results in a negative test simply because the immune system hasn't sounded the alarm loudly enough yet, not because the bacteria aren't there.
Exactly. You have to give the immune system time to react.
Because these Spirochetes are so insidious, so deeply woven into tissues, and so hard to pin down diagnostically, they haven't just caused physical medical problems. The source material shows they've been at the center of intense scientific debates and, in some cases, catastrophic ethical failures.
With syphilis, there is a long-standing historical debate regarding its origins. The sources detail the Columbian hypothesis, which argues that Christopher Columbus's crew brought syphilis back to Europe from the Americas in 1493. This supposedly ignited a massive, highly documented, and virulent outbreak in Naples in 1494.
Right, but there's a counter theory.
Yes, the rival pre-Columbian hypothesis suggests that treponemal diseases were already present in Europe, perhaps in a milder form, and either went misdiagnosed as leprosy or suddenly mutated to become more virulent. Both sides of the debate point to characteristic pitting and lesions left on ancient skeletal remains by the bacteria, interpreting the dating of those bones differently.
But there is a much darker, much more recent chapter in the history of syphilis research that reshaped medical ethics entirely. And the sources are very clear on the facts of this. The Tuskegee study of untreated syphilis in the Negro male.
Right. This was a longitudinal study run by the U.S. Public Health Service that lasted from 1932 all the way to 1972. The researchers enrolled roughly 400 black men in Macon County, Alabama, who were already infected with syphilis. The stated objective was to observe the natural, untreated progression of the disease over a lifetime.
The men were never informed of their actual diagnosis. They were told they were being treated for bad blood, a generic local term for various ailments.
And the most profound ethical failure occurred as the study progressed. Even after penicillin became the standard, widely available, and highly effective cure for syphilis by 1947, the researchers actively withheld the antibiotic from the participants.
They went to great lengths to prevent the men from accessing treatment through other public health avenues just to preserve the observational data of the study. And this continued for 40 years. It only ended because a public health worker named Peter Buxton acted as a whistleblower, leaking the details to the press.
Journalist Gene Heller broke the story in 1972, which sparked immense public outrage. The fallout from this discovery directly shaped modern medical regulations. It led to the creation of the Belmont Report, which forms the foundation of modern informed consent and the ethical protection of human subjects in research.
The history of these diseases is fraught, and there's a very real modern debate happening right now surrounding Lyme disease, specifically regarding chronic Lyme disease and post-treatment Lyme disease syndrome, or PTLDS.
The sources outline two distinct camps in the medical community on this issue. On one side, mainstream infectious disease bodies like the Infectious Diseases Society of America, or IDSA, maintain that a standard course of antibiotics clears the active borrelia infection.
But many patients continue to experience debilitating lingering fatigue, brain fog, and joint pain, right?
Yes. The IDSA views this as a post-infectious syndrome, essentially the immune system remaining in a state of chaotic inflammation long after the bacteria are dead. In their view, prolonged antibiotic courses offer no proven benefit and carry real medical risks, like disrupting the gut microbiome or breeding antibiotic resistance.
Okay, so what's the other side of the debate?
On the other side, a smaller community of clinicians, often organized under groups like the International Lyme and Associated Diseases Society, or ILEDS, argue a different biological mechanism. They propose that in some patients, the corkscrew bacteria persist, hiding deep in tissues where antibiotics struggle to reach, entering a dormant state. They argue this hard-to-detect active infection warrants extended, sometimes intravenous, antibiotic treatment.
I mean, for a patient feeling awful, being caught in the crossfire of this debate has to be incredibly isolating. The stakes are massive. As the sources point out, this dispute dictates whether insurance companies will cover specific treatments, it impacts malpractice exposure for physicians, and it deeply affects a patient's trust in the diagnostic process.
Which is exactly why understanding the root science of how these bacteria hide and operate is so vital for self-advocacy. The relevance of this microbiology cannot be overstated. These diseases are not historical footnotes. They are active, escalating public health issues.
Yeah, syphilis rates are climbing sharply across various demographics. Over the past decade, the numbers have surged, leading to a tragic rise in cases of congenital syphilis, where the infection is passed from an untreated pregnant mother to her newborn.
And the results of that are devastating.
It causes severe bone deformities, organ damage, and stillbirths. And it is a completely preventable tragedy with simple prenatal screening and basic penicillin.
Lyme disease is also expanding its reach. It is one of the most commonly reported vector-borne illnesses. As land use changes, suburban areas push into forests, and climate patterns shift, tick habitats are growing. More regions and more people are exposed to borrelia every single year.
So the details we've unpacked today, the 36 to 48-hour migration clock, the painless primary sore of syphilis, the complex serology tests, these things explain exactly why these diseases remain so tricky to manage today.
The shape of the spirochete dictates its survival. But before we finish, there is one final, terrifying trick in their playbook found in the research: antigenic variation. Neighboring spirochetes, like the one that causes relapsing fever, and to some extent the Lyme bacterium itself, use this mechanism to survive long-term.
What does that mean exactly, antigenic variation?
As they live inside a host, they periodically change their surface proteins. They literally alter the biological markers on their outer sheath.
Oh, wow. So they're constantly changing their molecular disguise.
Exactly. Just as the human immune system figures out what the bacteria looks like, produces the specific antibodies, and sends them out to destroy the invaders, the spirochete changes its coat.
The immune system is basically left holding a wanted poster for a disguise the bacteria no longer wears, and the body has to start the entire immune response over again.
It creates a continuous, exhausting cycle of immune evasion.
Which leaves you with a deeply unsettling question to chew on as we wrap up this deep dive. If a corkscrew bacterium can constantly change its disguise while burrowing deep into our gelatin-like tissues, will our immune systems, or even our oldest, most reliable antibiotics, eventually lose this microscopic arms race? Next time you see a corkscrew, just remember that the most relentless drills on the planet aren't in a toolbox. They are microscopic, they are ancient, and they might just be waiting in the tall grass.